Hi, I'm Jeff Barrett, and today I'm going to tell you about the Sanger Institute's comprehensive genomic surveillance of SARS-CoV-2 in the UK. Genomes can be used to study an infectious disease pandemic in a variety of ways. I'm going to start by just thinking about all of these use cases, considering the genome itself is just a barcode. So SARS-CoV-2 virus has a 30,000 base pair RNA encoded genome. It accumulates new mutations at a rate of something like one every couple of weeks. That's a bit slower than some other viruses that infect humans. If you think about that barcode, so it's just, you read it off and can use it as a label to track how the virus is transmitted from person to person and place to place, you can then deploy that in, broadly speaking, two different ways. One is in a targeted way, which is you focus on, say, a particular outbreak in a healthcare facility where you really want to find a specific set of individuals who might be patients or healthcare workers. You know a lot of metadata about them, so when they come on and off shift, how they move around, and you use the genomes to try to understand who's transmitting to whom and to differentiate between situations where, for example, there is a failure of infection control practice within the healthcare facility and individuals are transmitting inside versus multiple community-acquired infections making it into the healthcare facility. And the Sanger Institute's activities in SARS-CoV-2 sequencing are actually part of a larger initiative called the COG-UK Consortium, which brings together hundreds of different individuals in sequencing facilities, hospitals, virology experts, phylogeneticists, public health agencies who come together to use genomics in many ways, including a number of excellent case studies of this kind of targeted genomic epidemiology. I'm actually not going to talk really in any detail about that, although it has provided a super valuable resource, I think, in different ways of responding to the epidemic in the UK. I'm going to focus instead on the specific contribution that Sanger has made, which is using genomics in surveillance. And to do this, we really need to select samples in a very different way. So instead of these small number of specific samples, we want a high throughput operation that every week takes a random selection of samples who've tested positive for COVID and sequences those genomes as a way of just providing national surveillance. And again, we can use the genome as a barcode and look, for example, to detect a new outbreak where a single version of the viral genome suddenly spreads very quickly, which could be a signature, for example, a super spreader event. And if we can do that quickly enough, it might be the kind of thing that's useful alongside other kinds of data to help guide public health response locally. So to set this up, we need as I mentioned, a high throughput, steady state, week by week operation. And the Sanger established a collaboration with the Lighthouse labs, which are the large scale, small number of distributed PCR test facilities around the UK that most of the so-called pillar 2, so that's if you go to a car park or send away for a home test kit and take a swab and post it off, these are the facilities that do the PCR tests and give you your result back. And basically, we arranged for these chilled vans to every day go up and down the country to currently 5, soon 7 of these Lighthouse labs and bring many, many thousands of the waste material of these PCR tests down to the Sanger Institute for genome sequencing. And in fact, at the moment, we currently get about 500,000 of these samples in these vans coming every week. And they go into short-term -20 walk-in freezers that were set up on the Genome campus so that we can store something about like 6 or 7 million of these at any given time. These green boxes that the samples arrive in are full of 96 well plates that are essentially straight off the back of the PCR testing pipeline. We have to open up the boxes, take out plates, select a random subset of these every week, and then they go through a combined manual robotic facility, which first picks out just the positive test results. So what we get is a mix of positive and negative tests. So it picks out the positives onto consolidated plates, and then a subset of those are taken forward into sequencing every week. And we can currently pick out about 20,000 positive samples a week. At Sanger, we've been consistently in the latter part of 2020 running at between 5,000 and 8,000 sequences a week. We can also distribute additional positive samples to other parts of the CogUK network that I talked about. When they have capacity beyond whatever their local surveillance and targeted work, they can take some additional pillar 2 surveillance sequencing. And at present, actually, as of this week, that combined effort can do on the order of 10,000 new sequences every week that just randomly cover positive pillar 2 tests from around the UK. And what that has led is that if you look at the total number of sequences that the world has generated and shared publicly, the UK has done about half of that total, and in fact, just crossed 200,000 total sequences generated, quality control positive sequences generated, and about half of those come from the Sanger pipeline that I've just described, and the other half come from the other 15 or 16 distributed sequencing sites around the country. And that's a mix of, as I said, the surveillance and targeted work. But it really means that the UK has a very comprehensive genomic surveillance of the specific sequences of SARS-CoV-2 that are circulating around the country at any moment in time. Now at Sanger, the kind of question we wanted to ask for using that surveillance was to look in any given place week by week and see if we can detect essentially super spreading events where the same sequence is suddenly observed in a big puff of identical sequences at the same time, which suggests some kind of a local super spreader event that has generated lots of cases from a single source in a short period of time. And when we were retrospectively trying to see if this would work, we kind of looked back, and this is one particular place, York, a city in England. The light gray line is the overall flow of samples to Sanger over the course of the epidemic. So you can see the spring low level of testing, but there was a peak then, the quiet time in the summer, and then these data are now out of date, but the beginning of the peak in the autumn. And the solid black line is the equivalent in just York, and you can see it looks more or less like the UK average. The black dots are the weeks where we have enough sequences to do a statistical test of the kind I was just alluding to, where you simply ask how much diversity is there week by week in the empirical distribution across the whole country. And if you see a lot of homozygosity, that is the exact same sequence, you sort of turn on a light that says something interesting is going on here. Now, in the case of York, you can see we didn't have much data early on. We started to get in the fall to consistently having enough sequences to do this analysis. And if you peek at the data, this is one way of labeling each sequence that we had in York in that particular week. And these are essentially one way of summarizing those barcodes I was telling you about. And you can see that there's some diversity here. That diversity changes over time, and there's different kinds of sequences in the same place a few months later because mutations have arisen and because people move around and different versions of the virus circulating around the country. What we were looking for is something like this, which is a quite different picture. Again, there were cases in the spring, a peak in the fall, but an otherwise very unusual sharp peak in Swindon, in the south of England, in the summer. And this sharp peak was tied to a particular outbreak that happened in a logistics facility. And what you can see here is that the dots, some of them turned red, which means they've reached a statistical significance to say that there is elevated homozygosity, or essentially identical genomes, in these weeks. So you can see the signal is quite clear in this particular example, where almost all of the sequences that were chosen in Swindon in that week were exactly the same. That's quite different. And it looks again different if you fast forward a few months and see some diversity and the kind of echo highlighted here in orange of the same widely spread sequence that has accumulated some mutations, those additional digits that have been added onto the end over the intervening months. And what actually was happening in Swindon, this was sort of, became a fairly widely publicized event, was there was an outbreak in this particular workplace, which, as I mentioned, was a cold chain facility. So it's the kind of environment where super spreading has been observed to happen in many countries, similar kind of thing to what happens in meatpacking plants. And these are statements from the Swindon Borough Council as they kind of detected the outbreak in the facility, they tested in the facility, they designated Swindon in general as an area of concern. And unfortunately, and this isn't a criticism, but unfortunately just the response wasn't quite fast enough to prevent the outbreak from leaving this facility and circulating more generally in the community. And of course, what was really happening was there was this super spreading context that was even earlier that wasn't visible. And the point to just draw one's attention to is that the red dots from the genome data did begin to show a signal early enough, if you had the data fast enough, to have contributed, one might hope, to the response. And so if we can get this turnaround low enough, such that this is one element that goes to the public health agencies alongside everything else, maybe we can get these responses a day or 2 days or 3 days faster. And cumulatively, doing every response faster might mean that we can keep, when hopefully we get to another period of low transmission in the community, we can keep that low as long as possible. So we built a system that every week would basically just say, look around the country and run this analysis and say which places have the highest level of identical genomes, and just provide this as a live feed. And this was the live feed type work was built by Matt Sinnott, who's a postdoc in the group. And here's some data from the week to the end of November, for example. And you can see there's, in yellow is kind of, amber alert that there is some elevation, but it's not at the very top end of the distribution. And red is very high levels of homozygosity of identical genomes. And if you zoom in this particular one to the southeast of England, you can see this is the empirical distribution around England in that period. And you can see that Medway, this particular county in the southeast, is right out here in the tail. And indeed, Medway is bright red, and other areas in Kent, Canterbury, and other nearby areas are showing sort of elevated but not extreme levels. And we had actually seen a consistent signal sort of for several consecutive weeks in November in this part of the country, but we didn't quite know at that point what to do with it or how to best sort of communicate these alarms onwards to the public health agencies. And in fact, the Public Health England had noticed at exactly the same time a pattern that was also worrying in the same part of the country. So this is the same week, but now just considering the rate of new cases around the country, and yellow and green are kind of low, blue is medium, and darker blue and purple is high. And the end of November was actually the final week of the England month-long lockdown that had been running. That wasn't a strict lockdown like had been happening back in the spring. Schools were open. Many people were still going to work. There was some sort of circulation of individuals. So it had successfully brought cases down in almost all of the country. But Public Health England had noticed that it hadn't been as effective if we zoom in the southeast. There were high rates of cases right here in Medway, exactly the same place we had been looking. And indeed, if you walk forward 1 week to essentially the first week in December, now that lockdown had been lifted, that worryingly high rate had extended to essentially all of Kent and the southeast of England. And it was at this point that a separate team led by combined effort of Mira Chan's team, Public Health England, and other members of COG-UK, including Eric Foltz, Nick Lohman, and Andrew Rambo, had linked these two things together and said, actually, is it possible that this rapidly spreading sequence variant of the virus in the southeast is, well, they observed that it was strongly correlated with the overall increase in cases, and is it possible that there's a cause and effect relationship here? And it's actually quite a hard thing to prove, because exponential growth is a kind of counterintuitive thing, but in any given place where just, because maybe there's poor compliance with the lockdown, cases are increasing, there will often be some variant of the virus that happens to be sort of swept up on that wave. It surfs to being high frequency rather than it causing and driving that increase in cases. So there was a sort of flurry of activity in the middle part of December to try to understand whether this was a cause and effect relationship. One of those analyses was led by Eric Foltz and Neil Ferguson and others at Imperial, and really kind of built the statistical case. And in parallel with that was one that built on a collaboration that we at Sanger had just begun with Moritz Gerstung and Harald Wöhringer at EBI to combine a hierarchical model they had built of just the case rate place by place over time in the UK with our genomic estimate of the relative abundance of this new lineage, which was dubbed B117, versus all other variants or lineages of the virus. And this is just showing the red dots on each of these lines are the genomic estimate of the genomic observation of what fraction in each place was this new variant B117. So, and the red line is the kind of fit of the data, fit through the data of a spline. And you can see in the top, Medway, this place in the southeast I've been talking about, it was first observed in October and it had really ramped up to quite high frequency by November. And then if you look at, say, Milton Keynes, this is just a random example, in the middle of the country, it had started to increase in frequency by November, December. And then if you go further north in Manchester, by mid-December, it had just started being observed. So now we can couple these estimates of how much of this new variant is spreading with these blue dots are just the observed case numbers published day by day on the government website. And you can fit a model which says, okay, can I estimate what fraction of new cases are driven by this new variant versus attributable to other variants? And in Medway, you see that essentially it becomes very quickly the increase in cases is all this new variant. In Milton Keynes, the same thing happens. That as the new variant becomes prevalent, the case numbers go up. And in Manchester, the case numbers remain pretty low because, well, we don't yet know because, but the case numbers remain very low, and there has not yet been much observation of the new variant. And in fact, what you can do is more formally test essentially as the new variant is observed in each place, how well does it correlate with the growth in total cases? And in fact, both this analysis and the other one from Imperial that I alluded to show quite consistently that in fact, in each locality, as the new variant is observed, the case rates follow on and become more common. And that provides pretty convincing evidence that it is in fact B117 that is causally driving high rates of transmission rather than being a coincidence. And one way to think about that is to look at now splitting each local authority around England into the estimates of the R number for B117 on the left and the other lineages on the right. So the R number is how much the virus is growing or not over time. And if it's less than 1, that means the number of cases will shrink over time. If it's greater than 1, the number of cases will grow. So in this graph, blue is less than 1, red is greater than 1. And you can see that in the last week of lockdown, what was happening was on the right-hand side, mostly blue, other lineages were shrinking. The lockdown was broadly speaking working. On the left-hand side, B117 in all of the places it had got to was still growing. So the conditions of that November lockdown were insufficient to bring its R below 1. And then one has to remember that although the virus was causing us problems by evolving to a variant that is more transmissible, it's really what the virus does and what we do that combined essentially determine how cases grow. And after the lockdown ended, this is a couple of weeks further forward in early December, people were circulating and every variant was growing. So there was going to be a surge in cases in December regardless in England. But really, because B117 had now been seeded in many places and it was so much more transmissible, it rapidly was really exploding in the middle of December. Now, this meant that we have brought ourselves to the current situation where there's a very high number of cases, the healthcare system is stretched very thin, and it has led to, of course, a strict lockdown with schools closed in January in England. And the small piece of good news is if you look at the data from the first full week of that lockdown, both maps are broadly speaking blue. The other lineages are darker blue than B117, but it seems that the current lockdown is able to bring R less than 1 for all lineages, and that means that slowly the number of cases will go down, as now seems to be happening. And I think given that the rate of decrease is faster for other lineages, what will happen in coming weeks is essentially almost all other lineages will go extinct, and B117 and its descendants will be all of the infections in the UK in the next several months. So I've talked so far about tracking this new variant using the DNA as a barcode, but of course, the RNA in the case of this virus. Of course, it's not just a barcode, it's also the blueprint of how the virus functions biologically, and one can look at the specific mutations and try to understand what's happening in that way. And this has been done in targeted ways, and this is, for example, an example from malaria, where you can look for genes that predispose to drug resistance as they are observed, as drug resistance is observed. But you can also factor it into the surveillance approach, where you're looking for the arrival of new mutations that might confer, for example, escape to vaccines as they are deployed broadly. And this is the kind of thing that we had in mind that we thought would become relevant in 2021. In fact, it turns out to have become relevant in late 2020, because B117, this variant that was seen to now be spreading very rapidly, also had a couple of other factors that were observed during these analyses that made it very unusual. And the first is that it has a lot more mutations on the branch that defines it. And by that, I mean, we usually see, because this virus accumulates mutations relatively slowly, and in the UK we sequence lots, most of the part of the tree looks kind of like this box down here, where these short lines show that we observe mutations 1 by 1 and 2 by 2 as they are accumulated. But then the arrival of B117 from its most recently observed ancestor has this very long branch, which is very unusual. And in fact, another way to see how unusual that is to imagine mutations as a clock. So these are kind of new mutations accumulating on the y-axis and time passing on the x-axis. And these are other sequences in the neighborhood of B117. You can see that pretty much there's some noise, but as a straight line, over time from the beginning of the pandemic, new mutations accumulate. But when you look at specifically the B117 lineage, it again is accumulating in the time it's been circulating in a fairly linear way, but it had this big jump from the rest of the distribution. And so something happened that basically led it to arrive with 23 mutations compared to its most recently observed ancestor that's quite distinct from the pattern that we have usually been observing. And another thing that was added on top of the sheer number of mutations is what kind they were. And they were, in fact, a number of missense, so mutations that change the amino acid sequence of the proteins of the virus, and indeed a concerning number that were in the spike protein, which is the most important protein the virus uses to bind to human cells and is the target of all of the currently approved and late phase trial vaccines. So it's the kind of centerpiece of the human virus immune interaction. And so this always gets the attention of virologists and immunologists. And in particular, there were a couple of things, perhaps most notably this N501Y mutation, which is in the receptor binding domain. So really where the spike protein and the human ACE2 protein bind. And so this meant that this variant was spreading fast. It had a lot of mutations that appeared suddenly, and some of those mutations had prior evidence to suggest they might have a biological function. So it was really the combination of those clues that led the frenzy of work to try to understand what was happening with this new variant in late December. And it also was reminiscent of a totally independent observation that had been published. This is data from a paper in the New England Journal in late 2020, but there are very similar stories. Ravi Gupta's lab has one example in medRxiv, Avanzado et al. In Cell, where here's again a tree of some various sequences. And this is one particular patient who was infected with SARS-CoV-2 for several months and had various treatment and sort of relapsing, remitting amounts of virus over the course of this chronic infection. And you can see, first of all, that, again, there's a long branch where a bunch of mutations required, not in a branch that's suddenly seen out in the population, but in this one person whom they sequenced their infection over and over again. And in addition to lots of mutations, there were a whole bunch in the spike protein down here, and some of them were very similar to the mutations seen in B117. So N501Y is seen here. It appears late in the infection. This particular deletion in the N-terminal domain, N-terminal domain, is also almost identical to one observed in B117. And so it led there to be a hypothesis that these chronic infections create essentially the evolutionary opportunity for the virus to accumulate a whole bunch of mutations and possibly to sort of jump to a new fit state that can then possibly be transmitted on, that is immunologically somewhat distinct from the previous variants of the virus that we've seen. And then as things sort of started arriving thick and fast, there were two other similar stories that have appeared in the last month. So one was actually a variant now called B1351, or also 501Y V2, which is first observed in South Africa and seems to have spread very quickly there and has been observed now in other countries in the world. It had a similar kind of story, mutations in the receptor binding domain, including N501Y, and a large number of mutations on a branch. And then even more recently was this lineage that was identified in Brazil, in Manaus, the capital of the Amazonas state, which also has the same exact kind of picture, a long branch to its newly arrived lineage, so a large number of mutations, including several in spike, including N501Y. And this one has been observed in some travelers to Japan in that particular case. But basically, these new variants and these observations in chronically ill patients all have this similar kind of pattern. And although we can't prove it yet, it's a pretty consistent story that the huge number of infections that have happened around the world have given the virus a lot of chances to evolve. And certain environments, like chronically infected patients, give it a kind of opportunity to evolve in ways that we haven't really seen earlier on in the pandemic. And it's now happened several times, and we're going to have to really contend with these new variants in the next phase of the pandemic. I want to finally highlight some work from Inigo Martin-Corena at Sanger on how these things happened. And one way that these new mutations could have exploded would be a fundamentally different mutational process. These sort of bar charts show the kinds of mutations that happen. So C to T, for example, or C to A. And in the study of cancer, it's often useful to see different mutational processes will have a different kind of rainbow pattern on these pictures showing what is driving it. And you can see that this is basically basic SARS-CoV-2, and you get a lot of this C to T pattern in red here, and a little bit of this T to G in C to G in, C to T in the lower half of blue over here. That is what we consistently see. And then what we see in mutations that arrive after the B117 and B1351, which was the one first seen in South Africa, arise is essentially identical. So what this is showing is that it wasn't a fundamentally new mutational process that came onto the scene, that what's happened in each of these things is essentially the same thing that's always been happening. But if we ask specifically to look at the pattern of selection using a method called DNDS-CV that Inigo has developed, in this pair of bar parts here is the sort of older things. This is the B117 lineage after it emerged. This is B1351 after it emerged. They all show the fact that they're below this dashed line is essentially evidence of purifying selection. So this virus is pretty fit when it burst onto the scene in humans, and most of the time selection is just purifying new amino acid substitutions. But these bars, which are the mutations that defined the arrival of B117, B1351, and one of these chronically ill patients, all show evidence of brief bursts of positive selection. And they're sort of not statistically significant individually genome-wide, but actually if you look at this method allows you to make a statement about specific genes. In the case of spike, each of these particular groups of mutations does show evidence for positive selection. And what that means is that something happened that basically, again, allowed a new constellation of mutations to arise in each of these cases that seems to have given the virus a new set of biological properties and which we're now having to contend with. Okay, so to summarize, the genomic surveillance that we set up for SARS-CoV-2 in the UK wasn't exactly intended to find this kind of thing, but it did end up providing the infrastructure to detect this rapidly spreading B117 variant, and we alerted the world. And I think it's fair to say that in many countries, the same kind of thing could be happening, and they simply don't have systematic surveillance in place to detect this. That variant B117 has now spread throughout the UK. In the last data that I've checked, which is a few days old, it is now something like 86% of all positive tests around the UK. So it really is just taking over completely. As I mentioned, the strict lockdown that's currently in place in January 2021 does seem to be enough to bring its R number below 1. This variant has been observed in many countries. And again, because the amount of sequencing isn't that large, it is probably in other countries where it hasn't been observed and is probably at some frequency above that which people might be aware of it. And so I think it's actually urgent that in as many places as possible, restrictions are tightened to try to suppress the growth of this variant, which is, we think, something like 50% more transmissible than older variants. And I hope that other governments are able to kind of respond early enough to take advantage of that. Denmark is one positive story where, because they have quite good sequencing as well, they can see it, in December it grew from like 0.25% or 0.5% to 1% to 2% of all their cases. And that led them to make a decision to tighten their restrictions because they could just see that, the exponent was going to keep going if they didn't. As I just mentioned, there are other variants with similar properties that have been detected in the last month or so, and suggest that we're going to have to deal with these in the phase of the pandemic that's now happening in 2021. And because of that, we certainly intend to scale up and continue to operate the Sanger Lighthouse surveillance program throughout this year, because we think it'll be important to watch these variants that have been observed and to be on the lookout for new ones, for example, that might be involved in vaccine escape. Okay, I'll just finish by thanking, I tried to mention people in COG-UK more broadly and public health agencies. It's been a truly amazing collaboration with all of them. And within Sanger, there have been hundreds of people who have participated in this, in setting up the infrastructure, in processing these tens of thousands of samples, in building the metadata and analysis pipelines, in letting us keep this thing running week in, week out. Here are some of the key people, especially Dominic, John, Cordelia, Sonia, who have really been instrumental in delivering this whole thing. And it has been a real privilege to work with this totally amazing team, and has been, I think, kind of the defining aspect of doing science during this pandemic, is so many different people coming together with a goal of doing work and making a contribution to minimizing the effect of this pandemic. So thanks very much, and I'm looking forward to the discussion.